using disulfide linkages, which are given here using this red lines here. Okay, so these are the two red lines that we can see here. Now the important factor is that if we use papain, they will cleave just before these two disulfide linkages. As a result of that, as there are no linkages, no other linkages except for these two disulfide bonds, so they'll, after this cleavage before these two disulfide bond, lead to three different groups, actually three different parts. Two of the parts are of FAB portions and one part of this FC portions, right? Now if we treat the same type of protein, the same protein, with another protease which is called pepsin, which is a very common protein. If we digest it with pepsin, pepsin will cleave this protein in different region. It will cleave the protein just right after this disulfide linkages, which are present in the hinge region, and also it will cleave or segment the FC region in several different components. So what we get ultimately, we get small components of this FC portions, which we will find we get a very small components, as well as we get one large component which is having both the FAB portions, having heavy as well as the light chains. It is called the FAB prime 2 or FAB2 portions, because it is having two FAB segments. So using this kind of cleavages, if we run the gel, from those gel we can get the bands, and using the bands and using the marker we can get the molecular weight. Utilizing the molecular weight, we can actually determine what are the structure of these molecules. In the very beginning, when we haven't discussed or haven't learned the structure of this, using this kind of protein digestion techniques, we come to know the structure of this kind of molecules. Okay, now let us talk about another important concept. It is called isotype, allotype and idiotypic determinants that are present onto the surface or onto the surface of immunoglobulins, because they are proteins. Now we have talked about constant region, heavy chain, and the light chain, constant region, and all these things. Now isotypic determinant or isotypic determinant is something which is found to be common in a whole species. For example, if you look for a whole human, all the immunoglobulins of all human beings should carry a particular type of sequence similarity. And what is that? Now if we look at here, if we look in this picture, what we can see, this light blue color and this dark blue color, both these segments are of the heavy chain constant region, right? So heavy chain constant region, so CH, both this case CH, and this light color region is a constant region of the light chain, right? So we'll always be talking about constant region of light chain and constant region of heavy chain, not about the variable region. Now remember, in this case, what we can see in this, if we take mouse for example, two mouse immunoglobulin inside single mouse. Suppose we take a mouse and we took two immunoglobulin of the same mouse. What we get is that one of the immunoglobulin is having two of both of the immunoglobulins are having variable regions in this constant segment, because they are having some variations in their amino acid sequences, but they are similar. They are having similar kind of constant region of the light chain. So this part is called the isotypic determinant. Now if you take a same type or same species of animal like this mouse, you will must find this similarity between all the immunoglobulins of the all members of this particular species. So that's called the isotypic determinants. For example, if you take a human being, you can have variations in this CH region, but you should not have any variation in this CL region most of the cases, due to this kind of similarity among them, because they are very much closely related. Now for example, if we look about this allotypic determinants, in this allotypic determinants, what we get, we can get variations. Now in this allotypic determinants, we should get variations between you and me. For example, if we take so in this case we take a broad area, which is a whole species, so all human beings are having the same kind. In case of this allotypic determinants, it's much more related. Now what we can get, this allotypic determinant, is that this is much more related to genetic arrangement. Now as the genetic makeup of you is different than the genetic makeup of me, we will be having variations in our immunoglobulins. Suppose if you take a same species organism of the same species, one is you, another one is me. If we check both the immunoglobulins, this is for you and this is for me, what we get variations in the CH region, which is a constant heavy, as well as the constant light. Both this case we are having variations, because we are having different genetic makeup. Now if we are having if we are twin, then if we compare these immunoglobulins of two twin, then you shouldn't have this kind of differences, but otherwise most of the cases you should have this kind of differences. These are called allotypes. That means we both are